So now, why do we need enzymes for digestion? Think for a second about what you remember about enzymes. You remember that enzymes have, I always think of Pac-Man, that here's our enzyme, he has a very specific job, right? And then what can fit into here is called a substrate. The substrate is in our case of digestion, the macromolecule that we might be trying to break up. And so our Mr. Pac-Man will go ahead and eat the macromolecule, and he can break it down into two separate parts, maybe two smaller parts of the substrate. And what this does is that we can speed up chemical reactions that take place just by utilizing an enzyme. That is why enzymes are there. Within the body, we have our body temperature, which is about 37 degrees Celsius if you're not sick, and our digestive reaction rates are suitable for sustaining life for these required enzymes. So these enzymes can work at the body temperature without the need for raising the body temperature. So by utilizing enzymes at our body temperature, we can see chemical reactions take place at a faster rate. We don't have to raise the temperature at all. So enzymes typically will catalyze hydrolysis reactions, and so we are going to add water. And so by adding water, we can break apart the bonds between the macromolecules, and we can break them down into monomers. And that's what an enzyme can help do. Just to kind of wrap it up, our enzymes can help convert large macromolecules into smaller molecules that can be absorbed and used, which is the whole purpose of digestion. It just helps things happen at a faster pace. For example, we have amylase inside of our mouth, and we put stress on the bonds of glucose that hold together that starch molecule. And lastly, let's look at a source of substrate, the products from that substrate, and the optimum pH for some digestive enzymes. So the first one I want to talk about is amylase. This is an enzyme that will break down carbohydrates. Proteins we'll see being broken down by protease, and lipase enzymes will help break down lipids and fats. And so our example for amylase is seen within our salivary glands, and those are contained within our mouth region and kind of under your tongue. To break down proteins, we see the enzymes contained within pepsin. And to break down our lipids and fats, we have the example of the pancreatic lipase. Amylase's job is only to break down starch. Protease's job is going to break down polypeptide chains. And the job of lipase is to break down triglycerides that contain the glycerol as well as the fatty acid tails. So the product of what we see being broken down from the starch is going to be maltose. The product of our polypeptides is the monomer of amino acids, and there will be different amino acids contained. And then lipase will break down fatty acids and glycerol. And so the source of this, like I've said before, is the salivary gland. We will see our pepsin that will be released within the stomach. And the pancreas is going to release lipase. Our pH is a little neutral, slightly basic, 7 to 8 in the mouth from our salivary glands. Protease is like a 2 or a 3 within the stomach, and lipase is going to be at a 7.2 to 7.5. And so just to look at the details of amylase and how it's broken down, it's the hydrolysis between the bond of a 1,4 glycosidic bond that's going to be contained right here. So one of the carbons there is the first carbon, the other one is the 4 carbon. If water is added, that's where we begin to see the breakdown into maltose. Now the digestion of protein, like I said in our chart, is in the stomach at about a pH of 2 or 3, and we see the hydrolysis of peptide bond between the polypeptide chain that's going to help break it down into smaller peptides, which then will break it down into amino acids. And lastly, the digestion of lipids, which is within the pancreas, which is looking at lipase, or something called pancreatic lipase. So it requires the presence of bile salts to emulsify the lipid, and can also help increase the surface area of lipid for the digestion of fat. If we could break down those lipids, then that digestion of fat can actually take place easier. And so you'll see that the bile salts and pancreatic lipases will actually break down the glycerol head and the fatty acid molecules for better digestion.